Bismillahirrahmanirrahim. Assalamu alaikum. Hello and welcome to Biosciences with Shatreen Sawati. And today I'm going to deliver the lecture about the Wobble hypothesis. This is the second part of my first lecture about the codon, anticodon, and the codon degeneracy. So must watch my lecture about the codon and anticodon and codon degeneracy. Then you will be able to understand the concept, basic concept of the Wobble hypothesis. So now I'm going to start the lecture about the Wobble hypothesis. I will discuss the history, the comparison between the Wobble pairing and normal pairing, and the Wobble importance, and many more in this lecture. So now I am going to start my lecture with the history of the Wobble hypothesis. Wobble hypothesis was proposed by the Crick in 1966, and you are familiar with the Crick, Watson and Crick, who proposed the DNA model. According to the Crick, the base present at the 5 position of the anticodon binds with the base at the 3 position of the codon. I will show you how. But the base present at the 5 position on the anticodon is not complementary or not following the standard base pairing rule in the Wobble hypothesis. Is it clear? So this is the little bit about the history and the concept proposed by the or postulated by the Crick. So as you are familiar with the standard base pairing, that G pair with C and C pair with G means adenine, guanine, cytosine and thymine. I'm talking about the nitrogenous bases. So guanine with the cytosine, cytosine with the guanine, adenine with the uracil, and thymine with the adenine. So you can see here the DNA base as well as the complementary RNA base. So in RNA, as we are familiar that the uracil is present instead of the thymine. Now, what will happen? This hypothesis explains that in the codon-anticodon pairing, you know that the anticodon present the transfer RNA, which is responsible for the translation of the codon to pick up the specific amino acids from the cytoplasm for protein synthesis. So you can see this is the anticodon which is present in the transfer RNA, and you can see here this is the 5 end, and you count that from this direction to this. So this is the position number 1 of the anticodon. And if you see here, this is the position number 1, 5 to 3 direction. This is the position number 1 of the codon. So as we are familiar that the codon and anticodon both are complementary to each other. But what will happen in the Wobble hypothesis? So you can see this is the position number 1 of the anticodon at the 5 end, and this is the position number 3 of the codon at the 3 end. Both this first base at the anticodon and the third base at the codon, they are not complementary with each other. These two are not complementary with each other. So this is actually the Wobble position or Wobble hypothesis. Is it clear? This Wobble position do not follow the standard standard base pairing. Like here's G. G must be complementary to C. But in Wobble pairing, what will happen? G is complementary to C as well as U. Now this will help the anticodon to recognize the more than one codon. As you are familiar with the codon degeneracy, that there are many more amino acids which can be encoded by 2, 3, 4, 5 codons. So this Wobble position help in what? Recognizing the number of the codon which are responsible for the protein synthesis, or you can say the picking of amino acids from the cytoplasm. So what will happen? The position 1 at the anticodon is not complementary to the position 3 of the codon. Is it clear? Means this position, Wobble position, do not follow the standard base pairing, and this is actually the Wobble hypothesis, and the position is known as the Wobble position. The concept clear now? Okay. Now you can see here the normal pairing comparison with the Wobble pairing. Here, this is the leucine. This is the amino acid. This amino acid, according to the degeneracy, codon degeneracy rule, is encoded by CUC, this codon and this codon. Means the two codon. You can see here the two codon are responsible for the same amino acid, which is explained in the codon degeneracy. So what will happen? This is in the normal pairing. What will happen? G is with C. But in the Wobble pairing, what will happen? The G can recognize the codon with the U at the third position. Is it clear? Here at the third position, C is present. This is the normal pairing, standard base pairing. But this is the Wobble pairing. In this case, G can recognize the U. Means the G can recognize the codon with the U at the position number 3. Is it clear? So this is the comparison between the normal and the Wobble pairing. Okay. Now I'm going to discuss about the Wobble positions. I will show you here the 3 basic concepts related to the Wobble position. Number 1. In number 1, what will happen? At the anticodon, at this position, position number 1, which is the Wobble position. Now I call this position is the Wobble position because now the concept is clear upon you. So this Wobble position, number 1, base at the anticodon, can recognize the codon with G, A, U and many more. So what will happen? The first case. In the first case, these are the cases related to the Wobble position. In the first case, what will happen? The presence of the CRA at the position number 1, at the position 1, at the anticodon, can recognize just one codon. Is it clear? So what will happen? If the CRA present at the first position of the anticodon, then this type of the anticodon can recognize just one codon. This type of anticodon can recognize just one codon if the CRA present at the Wobble position. Second case, if the U or G, you can see here U is present. So you can recognize the codon ending with G or U. Is it clear? We are familiar that U is complementary to A, but here you can also base pair with the G in the Wobble hypothesis. Is it clear? So you can recognize if the U is present at the Wobble position, then it can recognize the G as well as A of the codon. Mean it can recognize two codon. Similarly, if the G is present, then it can also recognize the two codon ending with C and U at the position number 3 of the codon. I hope so the concept will be clear upon you people. Okay. Now the third case. Most of students are confused about the I. What is I and what is the role? So I is for inosine. If the inosine base is present, this is also the nucleotide. If this nucleotide is present at the position 1 of the anticodon, then it can recognize 3 codon. You can see here the 3 codon 1, 2, 3. If the inosine is present at the position number 1 of the anticodon, means the Wobble position, so it can recognize the 3 codon like you can see here, it can recognize the codon with the A at third position. It can recognize the codon with C at third position, Wobble position. And it can recognize the codon with U at the Wobble position. But what will happen if the inosine is present at the position number 1 of the anticodon? Is it clear? So this is explaining the codon degeneracy. Here you can see just one anticodon can recognize the 3 codon for the same amino acid alanine. Is it clear? Inosine is able to form the bond with the adenine, with the cytosine, with the uracil. Must note down this. Inosine can form bond with the adenine, cytosine, and uracil. So if the inosine is present at the position number 1 of the anticodon or the Wobble position, then it can recognize 3 codon for the same amino acid. That is explaining the codon degeneracy. Is it clear? Okay. Now, what is inosine? Inosine is a nucleotide that is formed when the hypoxanthine, as you are familiar that the adenine is present at the position number 1 of the ribose sugar, and guanine, cytosine, thymine, uracil. You are familiar with the normal nucleotide. But this is not the normal case. This is the Wobble case. In this case, the nucleoside inosine is formed how? When the hypoxanthine is attached to the ribose ring, also known as ribofuranose. And the bond between the hypoxanthine and the ribose is the beta nitrogen 9 glycosidic bond. Beta N9 glycosidic bond. Is it clear? Do you have the concept of glycosidic bond? If you don't have the concept, must watch my lecture related to that. Glycosidic linkages. So, inosine is commonly found in the transfer RNA and is essential for the proper translation of the genetic code, means the code response for the codon degeneracy in the Wobble base pairs. Is it clear? Okay. Now the role of inosine. Why it is important and how the inosine modification occur and at what stage of the transcription. The nucleoside inosine play an important role in purine biosynthesis. Must keep it in your mind. Inosine is responsible for purine biosynthesis, purine, adenine, and guanine. Is it clear? And also for the gene translation, modulation of the fate of the RNAs. Now, how the inosine is produced or editing of the transfer RNA occur. The editing of the transfer RNA with inosine occur at the post-transcriptional stage. This will occur, this modification occur, and this modification is responsible for the editing of the adenosine to inosine. Must keep it in your mind. This editing is responsible for the adenosine to inosine, and it occur at the post-transcriptional stage in transfer RNA, and also it is found in the messenger RNA. But Wobble hypothesis related to the to the modification in transfer RNA. Is it clear? At the Wobble position of the transfer RNA anticodon, inosine profoundly modify codon recognition. Means if inosine is present, it can recognize the 3 codon, and now you are familiar with the inosine can bind with or pair with the you can see here adenine, cytosine, as well as uracil. So it can recognize the 3 electrons for the same amino acid. Is it clear now? Now there are many other functions related to the inosine. While in messenger RNA, inosines can modify the sequence of the translated polypeptide or modulate the stability, localization, and splicing of the transcript. Must keep it in case of the messenger RNA. Is it clear? Now, inosine is also found in non-coding and exogenous RNA, where it play a key structure and function role. In addition, molecular inosine is an important secondary metabolite in purine metabolism, as I have discussed, that also act as a molecular messenger in cell signaling pathway. And I will share the link related to the inosine and inshallah in my description box. If you have still any question, you can ask me. I will try my best to answer you people, and I will be back with a new lecture soon, inshallah. Thank you very much, and till then, Allah Hafiz. You can also read the Wobble hypothesis importance. Basically, I have discussed in detail that the Wobble hypothesis help in the codon degeneracy. So thank you very much, and Allah Hafiz.